Hi everyone, my name is Pranali, aka your Lab Code Girl. Today I'm back with another video on a very hot topic, and that is contractile vacuoles. So I'll be majorly focusing on contractile vacuoles, which are present in algae, amoeba, and a yeast cell. Now before getting further with this video, I just want to make an humble request to all my viewers that if you all are watching the video and if you find it to be really helpful, please do consider liking if you do genuinely. And also, please do not forget to hit the subscribe button because that will actually encourage me and motivate me to create more such videos and to upload them here as well. Now, before getting any further ado, let's just get into the video. So, today I'll try to throw some light on contractile vacuole, as I already said, majorly in algae, amoeba, and vacuoles present in yeast cells such as Saccharomyces cerevisiae. Before I talk about the significance of the vacuoles in this organism, let's have a look at this very, very simple definition, although the structure is quite difficult. The contractile vacuoles are nothing but a membrane bound organelle which are mostly present in the members belonging to the kingdom Protista, such as algae and amoeba. Also, I thought it's a good idea to revise a little bit about the organisms of our interest as well. So, I'll begin with algae, and I'm sure that each one of us are aware of seaweeds or algal blooms by all means, right? And these are nothing but an algae. So, these are defined as a group of predominantly aquatic, photosynthetic, and a nucleus bearing organism that lacks the true roots, stems, and leaves. Moving on to amoeba, it's a genus belonging to protozoa, which are unicellular eukaryote with membrane bound cell organelles. I would like you to remember that it's found in terrestrial as well as aquatic habitats. And later in the video, you'll come to know why. The cell of an amoeba consists of a cell membrane, nucleus with a genetic material, food vacuole for digestion, contractile vacuoles for osmoregulation, and finally, the last organism of interest is definitely yeast. These are single celled fungus known to ferment sugar to produce alcohol and carbon dioxide. Now have a good look at all the three images, which actually depicts a schematic and representation of all the three organisms, which I revised just now. Now observe majorly on the organelle part. I hope the image is clear to you all. If it doesn't, I'll just try and read it out. Now if you look at the algal cell, it does contain cell wall, cell membrane, nucleus, endoplasmic reticulum, Golgi apparatus, mitochondria, and many more. And if you look at the yeast cell particularly, it also does have cell wall, cytoplasm, cell membrane, nucleus, Botska, mitochondria, vacuole, lipid granule, and etc. And when we look at amoeba, just focus that it is devoid of a cell wall. It do contain cell membrane. It also has cytoplasm, which is divided into two parts, ectoplasm and endoplasm. It also has pseudopods. It also has nucleus, nucleolus, water globules, etc. Yeah, the main interest. It also has contractile vacuole. Now, as we know that in certain organisms, such as single-celled organism, lacking the cell wall, usually contains an organelle called as contractile vacuole. Now, a vacuole works just the same as its name suggests, in that it expands and contracts. The point of the contractile vacuoles is to pump water out of the cell through a process called as osmoregulation. Now, it occurs in freshwater protist, but mainly in the kingdom Protista as a whole. Now, let's understand how it works. So, simply, there is need to be a set of amount of solute versus solvent in a cell. Basically, the solvent, which is water, needs to be in balance with the other solute, for example, salt. Now, since the cell works by osmosis, the water moving across a cell membrane, which is semi permeable in nature, as we all know, it is important to know that water always moves to a lower concentration of a water and a high concentration of the solutes. Now, if we look at the freshwater environment, they will contain comparatively less salt, than in any marine environment. In this condition, the organism cell will contain more solute concentration than in the external environment. Thus, we can say that concentration of the solute is hypotonic in the external environment. Now, under this condition, due to osmosis, the water will tend to accumulate in the cell from the external environment, and here comes the role of contractile vacuole, that it acts as a part of a protective mechanism, that it prevents the cell from absorbing too much of water and possibly lysing or rupturing through excessive internal pressure. Now, having been said that so much, I expect that you can now answer this very simple question, that is, why does not an amoeba burst like a red blood cell? Now, I'll pause here for a second or a minute so that you can just write down the answer or think about an answer. It's well and good if you can actually answer this question, and never mind if you didn't, because I'm anyways going to say the answer and you can just cross check yourself whether you're right or wrong. Ta-da! The time is over. Now, the simple answer to the question is that amoeba has contractile vacuoles that absorbs the excess water and it releases to the outside environment, hence preventing the bursting. Unlike the red blood cell here in the picture, you can see the steps from 1 to 7. How does it happen? Now, first, when you look here, the water is entering the cell due to the osmosis, and then the excess water will enter the contractile vacuole. Then the contractile vacuole will swell just like a balloon. When we blow an air into it, it just gets swells. And then the contractile vacuoles, which is filled with, you know, it's filled with the complete water. It will try and move towards the edge of the cell, and when the contractile vacuole is filled, now it has to be empty. So it expels the water by getting bursted, and then this cycle is repeated again and again. That is how the cycle continues. Now, here we will look at some important terminologies used more often regarding the contractile vacuole, such as systole and diastole. Now, I'll just read the points from here. Even you can read along. The contractile phases of the collecting water, that is expansion, and expelling water, that is contraction, are periodical in nature. Now, one cycle takes several seconds depending on the species and the environment's osmolarity. The stage in which the water flows into the contractile vacuole is called as diastole, and the contraction of the contractile vacuole and the expulsion of water from the cell is called systole. Water always flows from outside the cell into the cytoplasm and only then from the cytoplasm into the contractile vacuole. The amount of water which is expelled from the cell and the rate of contraction are related to the osmolarity of the environment. And in the hyperosmotic environment, the less water will be expelled and the contraction cycle will be longer in nature. I hope we all are good with the contractile vacuoles in algae and amoeba. Now it's the correct time to learn about vacuoles which are present in yeast cells, unlike the contractile vacuoles which we saw in algae and amoeba, whose major function is just osmoregulation. But these vacuoles are highly dynamic in nature, and these are also highly regulated as well. As I had previously tried to define yeast cells, I mentioned that this underlies in a kingdom fungi, and therefore we can call yeast cells, the yeast vacuoles, as a fungal vacuoles too. And these are acidic organelles with degradative and storage capabilities that have many similarities to the mammalian lysosomes and also plant vacuoles. Now, in past several years, well-developed genetic, genomic, biochemical, and cell biological tools in Saccharomyces cerevisiae have provided fresh insights into the vacuolar protein, organelle acidification, ion homeostasis, autophagy, and stress related functions of the vacuole, and these insights have often found parallels in mammalian lysosomes. Now, as I said, that vacuoles in the yeast cells are unexpectedly dynamic in nature and it is highly regulated. It is also considered as the last compartment of the biosynthetic pathways and the endocytic pathways, that is, the garbage dump of the cell. Hence, it is really difficult to make you understand, and therefore, I simply put across this image to make it easier and understandable. I hope this will make sense once I explain this figure to you all. Don't worry, it's very, very simple. So, I'll begin. Now, majorly, this vacuole is responsible for multiple constitutive processes like degradation, storage, buffering, and detoxification. Sorry if you think that I'm going slow over here, because I'm purposely going slow because I just want you to understand the function which is really important of this contractile vacuoles in ESL. So I'll be probably going very, very slow. So yeah, coming back, I'll explain all this multiple constitutive function by going in a sequence. Now if you see here this yellow color circle, which you can see, these are enzyme hydrolases. The vacuole is highly enriched for this, that eventually breaks down the cargo which is delivered in a multiple trafficking pathways to the vacuole. The second function which I mentioned is for storage. Now this vacuolar membrane contains multiple transported, this blue color ones. These blue color polygons which you see here are the transporters that imports the amino acids, ions and the metal. Now, the acidic nature is, if you remember, I've already mentioned about this. This acidic nature is conferred to this vacuole is due to the presence of a vacuolase H plus ATPase enzyme. This, the green color one, and this is required for the proper maturation of this yellow color hydrolases and for establishing the proton gradient that drives the many transporter. Now, the third function which I mentioned was buffering. Now, the vacuolar membrane not only consists of importers, but it also consists of an exporter as well. And these are here. You can see by the arrow, these are exporters, this pink color one or a lilac color ones. These transporters that recycle the amino acids, ions, and metals to the cytosol. Now, the combined activity of the importers and the exporters, this blue color one and this pink color ones, leads to the maintenance of the ion homeostasis and the amino acid recycling. Now, the vacuole is the main storage site for the polyphosphate, which buffer the cations and is a source for cellular phosphate. And the last function I said about detoxification. The vacuole also sequesters some toxic materials uh, such as toxic metals and potential harmful metabolic byproducts via ABC transporters. So it has to remove those by, and that is why its main function is also in detoxification. I hope the function is clear. Now it's also very important phenomena to look upon is how does a vacuole responds towards certain stress such as nutrient deprivation or upon a salt stress, etc. If there is a nutrient deprivation, meaning there's a less nutrient present in an external environment. Now, multiple autophagic pathways deliver specific and non-specific cargo that is organelle to the vacuole. Now, there are two types of autophagic pathway. The first one is macroautophagy, in which the non-specific degradation occurs of the cytosolic proteins and the organelle. The autophagic membrane originate from pre-autosomal structure called as PAS that enclose the cytosolic organelle, forming the autophagosomes, which are then delivered to the vacuole. And then the second one is microautophagy. Now, in this, the vacuolar membrane invaginates and directly engulf the cargo for the degradation. Okay. And then here, which you see is PMN, stands for piecemeal microautophagy. It's a type of autophagy that specifically degrades the nuclear components only. I'm so sorry if you can't see the cursor. I think there's some issue with this recording. So please just try and adjust with this. And I'm really sorry again. I hope this issue doesn't pop up next time. You can see over the image as I'm saying, you can just refer over here. Yeah. So, yeah, I'll talk about pixophagy now. This degrades the peroxisomes via two mechanisms similar to macro and the microautophagy. Now, when there is a salt stress, these vacuole fragments into multiple tiny vessels eventually, leading to the activation of certain importers at the vacuole to overcome the stress which is caused. And these are few major references, though I've referred a lot more since the same information was repeated. I chose not to incorporate all those as such. And these are few websites, renowned websites, and this is one of the research paper which I referred to get information about yeast vacuoles in Saccharomyces cerevisiae, because I when I was actually looking for videos, uh, I had to give a presentation in my college. So just to prepare myself, I was looking, since I did not get so much information about yeast vacuoles on any website particularly. Then I was looking. Then I came across this proper research paper. So I thought that I'll just refer this, and I this is really a good one actually. I'm so sorry for blabbering. Yeah, thank you so much for your kind attention and time. Have a nice day ahead. And also, please, please, please do subscribe my channel, and also consider liking it if you do it generally. Okay, I'm so sorry. I'm just